Hi children, let us continue our discussion for the principles of inheritance and variation, and we will start this video for the explanation of the concept of dominance. यहाँ देखिए कि genes जो है वो characters को control करते हैं, that we know, and genes, they are nothing but the segments of DNA, right? And DNA, it is the genetic material. Each gene has two alternative forms which are called as alleles. And let us take an example of a gene which is responsible for the production of an enzyme. And enzyme का काम हम जानते हैं, it changes the substrate into a product, right? Enzyme in the living beings, what it does, it enhances the rate of biochemical reactions, right? It actually manipulates the rate of biochemical reactions which are taking place inside the living being. So actually they are what? They are the proteinous substances which changes the substrate into product. All in all, they are the biocatalyst. However, what we are discussing here, हम यहाँ पर genes के रोल को डिस्कस कर रहे हैं. जीन्स, यहाँ पर हमने एग्जाम्पल लिया है कि जीन एक एंजाइम के प्रोडक्शन के लिए रिस्पॉन्सिबल है. अब देखिए इसकी जो ऑल्टरनेटिव फॉर्म्स है, राइट? सो वन, इट इज व्हाट? इट इज अनमॉडिफाइड वन. व्हाट? वन अनमॉडिफाइड एलील. बिकॉज एलील्स जो है वो मॉडिफाई भी हो सकती हैं. व्हाई इट कैन बी मॉडिफाइड? As you know, the replication of DNA, it is a biochemical process, and no biochemical process, it is 100% accurate, right? So the alleles, which are the alternative forms of the genes, they can be modified as well, right? And that modified allele can produce the different phenotype. ये चीज रिस्पॉन्सिबल होती है डिफरेंट कैरेक्टर्स के अपीयरेंस के लिए. नहीं तो एक ही कैरेक्टर रीअपियर होता रहे, क्योंकि सेम जीन, इट इज ट्रांसफर्ड फ्रॉम वन generation to another. So the appearance of the different character, it is because of the modification in the alleles, modification in the genes. Why? It can be modified. कोई ऐसा restriction नहीं है modify नहीं हो पाए, तो it can be modified. सो अब एक जीन का हमने एग्जाम्पल लिया, दैट इज रिस्पॉन्सिबल फॉर द प्रोडक्शन ऑफ एन एंजाइम. एंड एंजाइम का आप ऐसे समझ लीजिए कि स्पेसिफिक रोल है किसी केमिकल रिएक्शन को एनहांस करने के लिए, किसी सबस्ट्रेट को प्रोडक्ट में चेंज करने के लिए. यहाँ पे आप देखिए अनमॉडिफाइड एलील जो है वो डोमिनेंट कैरेक्टर के लिए रिस्पॉन्सिबल है. इट इज रिस्पॉन्सिबल फॉर द डोमिनेंट कैरेक्टर, राइट? अब जो मॉडिफाइड एलील है, सपोज एक एलील जो है वो मॉडिफाइड हो गया. सो अब मॉडिफाइड एलील क्या क्या कर सकता है? आइदर इट प्रोड्यूस लेस एफिशिएंट एंजाइम, और नॉन फंक्शनल एंजाइम, और नो एंजाइम. क्योंकि आप देखो कि जीन का, एलील का स्ट्रक्चर चेंज हो गया, राइट? सो उसकी इंफॉर्मेशन भी मैनिपुलेट हो जाएगी. एंड किस तरह से होगी कि आइदर इट इज रिस्पॉन्सिबल फॉर द लेस एफिशिएंट एंजाइम, फॉर द प्रोडक्शन ऑफ लेस एफिशिएंट एंजाइम देन फॉर्मर, और इट इज रिस्पॉन्सिबल फॉर द नॉन फंक्शनल एंजाइम और नो एंजाइम. अब देखिए इस केस में, जबकि जो मॉडिफाइड एलील है वो एक लेस एफिशिएंट एंजाइम के लिए रिस्पॉन्सिबल है, तो क्या होगा कि एंजाइम की एफिशिएंसी तो कम हो गई लेकिन काम चलता रहेगा. तो इसका मतलब कि सेम कैरेक्टर इट इज शोन. What the former responsibility of that particular gene is, it is carried, and here the same character it is shown, like that of which is good in number, which can be said to be dominant, right? So the same the same character it is carried. Now अगर नॉन फंक्शनल एंजाइम है या फिर जीन जो मॉडिफाइड एलील है, इट इज रिस्पॉन्सिबल फॉर द नॉन अपीयरेंस ऑफ द एंजाइम, कोई एंजाइम नहीं बना, तब क्या होगा कि इंफॉर्मेशन चेंज हो गई, एंड दैट रिजल्टेड इन द डिफरेंट फिनोटाइप. डिफरेंट फिनोटाइप्स दिखेंगे, बिकॉज द structure is changed so as to information, right? So नेचर में ये केस ज्यादातर होता है कि अगर एलील अगर चेंज हो भी गया, राइट, उसका स्ट्रक्चर मॉडिफाइड हो गया, तब भी एफिशिएंसी कम हुई लेकिन काम चलता रहा, तो वो कैरेक्टर अपियर होता रहा। लेकिन अगर फिर ये दो केसेस हैं, तो फिर जो इंफॉर्मेशन है वो चेंज हो जाएगी, और इट रिजल्टेड इन द डिफरेंस different phenotype, you can say, and that is why the recessive character it appears, right? So this is the reason the dominant character is there, it is good in number, but despite of the fact that dominant it is there, it is good in number, the other characters in terms of recessive character it is also there, and that is because of the modified allele. And the modified allele, it in case of recessive character, it is expressed in such a way where the structure of the allele it changes, and so as to phenotype, structure change हो गया तो information भी change हो गई और the phenotype भी change हो गया, और इस तरह से हमें recessive character देखने को मिला. So this is the explanation of the concept of dominance. That एक बार फिर से देख लीजिए, जीन्स जो है वो कैरेक्टर्स को कंट्रोल करते हैं, एंड एक एक जनरेशन से दूसरी जनरेशन में पास ऑन होते हैं. एलील्स, they are the alternative forms of gene. Alleles can be modified, in case you can say, as the example of heterozygous individual, where the alleles they are different, right? So alleles can be modified, and if the modification of allele it changes the information, then the phenotype it also changes, right? And this is the basis of the appearance of the recessive character. So ये explanation of the concept of dominance था, जिसे मैंने समझाने की पूरी कोशिश की है. अगर कोई चीज़ समझ में नहीं आए तो you can comment in the comment box. Now come to the another topics of this chapter, that is codominance and multiple allelism. चिल्ड्रन, इससे पहले वाले वीडियो में मैंने आपको बताया इनकम्प्लीट डोमिनेंस, और इनकम्प्लीट डोमिनेंस जो है वो यू नो its name indicate everything here. The dominance is incompleted, it is intermediate in between the dominant and recessive character. Now I'm not going to revise this because you can see the incomplete dominance in the previous video, but still we can take the example of a snapdragon where the incomplete dominance it can be seen, and here in place of red flowers the pink flowers they appear, and the phenotypic and genotypic ratio in the F2 generation, that is 1:2:1. Now come to the codominance, and codominance can be seen in the ABO blood group system. You know, the four types of blood groups they are there: A, B, AB and O. The plasma membrane of the erythrocytes or the RBC, they are having the sugar polymers which are protruded from it, and the alleles they are responsible for the different types of sugar, slightly different types of sugar. So gene that is I, right? Gene which is responsible, that is I, and the three types of alleles they are what? They are IA, IB and I, right? The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar it is controlled by the gene which is what? Which is I, and the three alleles are IA, IB and I. Now ये कैरेक्टर थ्री एलील्स के थ्रू कंट्रोल होता है, लेकिन आपको पता है कि एक डिप्लॉयड ऑर्गेनिज्म में ओनली टू एलील्स कैन एंटर, राइट? एक diploid organism में only two alleles can enter, and that is why the combinations are there. See, when IA it comes with IO. You know, this small I, it can be written as IO as well. So you can take IO also. In NCRT, small I it is given. So don't get confused for that. Here, when IA it comes with IO, then what blood group will be? It is A. When IA it comes with IA, then blood group will be A. BIO, then B. B with B, then B. A with B, then AB. And O with O, like the same alleles, small I or IO, then O. So that is how the blood groups they are controlled by the different combinations of the alleles. Here, what you can see through the description of this, that IA and IB they are dominant over IO, which can be written as small I as well. They can be dominant over IO, and when IA it comes with IB, then both show their effect and the AB blood group it is formed. No one can dominate other. In this combination, no one can dominate other, and AB it is produced. This blood group it is produced. Other than when IA it comes with IO, then IO is recessive, IA comes, और जब IA के साथ आया तब तो कोई कहना ही नहीं है, तब तो A हो गई होगा, right? So that is similar to this one also. IB, when it comes with IO, then B, because it is dominant over this, and IB, when it comes with IB, then B. A, when comes with B, then AB is produced, and IO, when comes with IO, then O is produced. So ये significant है, ये combination. Why it is significant? Because no allele can dominate the other, right? And that is how the AB is produced, and this phenomena is known as codominance. This is codominance. Codominance is when the heterozygote shows the traits associated with both homozygous genotypes, right? Often you see codominance in molecular traits. And here the AB blood group, it is the example of the codominance where the IA and IB both shows its effect. Now, one more phenomena it is associated with this, that is multiple allelism. And what the multiple allelism is. First of all, we have to clear with alleles. ये मैं आपको कई बार बता चुकी हूँ. Alleles are nothing but the alternative forms of genes, because genes they are there to control the characters, and the alleles are the forms, right, for the like dominant character or recessive character. So alleles they are nothing but the alternative forms of genes. यहाँ पर क्या important है, यहाँ पर क्या अलग है, that single gene is having three alleles or only one character, that is the type of sugar, right, of the plasma membrane in the RBC, that is controlled by these three alleles IA, IB and IO, which can be written as small I as well. So this phenomena is known as multiple allelism, where the character is controlled by more than two alleles. More than two alleles. In general, the two alleles are there, either dominant or recessive, right? In the Mendel's experiments, we have seen this either dominant or recessive. But this is somewhat different where a character is controlled by more than two alleles. And here you can see one gene is having the three different alleles, or you can say one character is controlled by three different alleles, and this phenomena is known as multiple allelism. So here the ABO blood grouping system, right? Here the two phenomena they are being shown, what the codominance and the multiple allelism. And here let's come to the another topic of this chapter, that is chromosomal theory of inheritance. But before the start of this description, I will like to clear here that sometimes a gene product is responsible for the controlling of more than one phenotypic trait. How? See, this is also a different case. As we know, children, that genes they control the characters. अभी तक हम यही देखते आ रहे हैं कि जीन्स जो है वो कैरेक्टर्स को कंट्रोल करते हैं एंड there is no doubt in that dominant character is controlled by dominant gene, the recessive character is controlled by the recessive gene. यहाँ पर एक एग्जाम्पल दीजिए, that is P, and P seeds, इनमें स्टार्च ग्रेन की सिंथेसिस को एक जीन कंट्रोल करती है, that is capital B and small b, the combination, like the gene is capital B and these are the two alleles, you can say capital B and small b, small b. Now capital B is responsible for the synthesis of the long starch grain, and small b, it is responsible for the short starch grain. Right? Now, let us discuss the cross here. ये जीन एक और phenotypic trait कंट्रोल करती है, that is the shape of the seed. And here the round shape, it is controlled by capital B, and wrinkled shape, it is controlled by the small b. Now, when we can make a cross here in the first filial generation, what is obtained? The heterozygote, it is obtained. Heterozygous condition should be there. And in the F1 generation, all the seeds they are round in shape. So you can easily say that round character, round seed shape, it is dominant over the wrinkled seed shape. But what about the other phenotypic trait which is also controlled by this gene, that is the formation of a starch grain? Here the size of a starch grain of this heterozygote it is what? It is intermediate. होना क्या चाहिए था? होना चाहिए था कि long starch grain should be there, right? Because in general आप सोच सकते हो कि that should be dominant over the short starch grain, but here the intermediate size it is obtained by the heterozygous condition. So here you can see that a single gene product, it is there to control the two characters. What? First of all, the synthesis of starch grain, and second is the seed shape. और उसमें भी दो डिफरेंट रिजल्ट्स हैं, राइट? हेयर सीड शेप के केस में राउंड इज डोमिनेंट ओवर रिंकल, बट अगर स्टार्च ग्रेन की आप बात करोगे तो स्टार्च ग्रेन के केस में यहाँ पर इंटरमीडिएट साइज आ गया, ना लॉन्ग ना शॉर्ट, बीच में इंटरमीडिएट। एंड हेयर जब हम स्टार्च ग्रेन के सिंथेसिस की बात कर रहे हैं तो यहाँ पर क्या शो हुआ, यहाँ पर इनकम्प्लीट डोमिनेंस शो हुआ, right? Interesting case it is. So occasionally a single gene product may produce more than one effect, and here the example I have given you, right? Starch synthesis in pea seeds, and it is controlled by the gene along with the seed shape as well. So you can say here, children, that actually the dominance, it is not an autonomous feature of a gene or the product that it has information for. What it is depended on, it depends as much on the gene product and the production of a particular phenotype from the product as it does on the particular phenotype that we choose to examine, right? So आई थिंक ये आपको समझ में आ गया होगा। अगर आपको ये वीडियो अच्छा लगा है तो लाइक करिए, शेयर करिए और आप अपनी रिक्वायरमेंट्स भी कमेंट कर सकते हैं। बाय फॉर नाउ।